Hi, everyone. As Mariceli said, thank you. I could jump in here. I'm a crop disease geospatial data scientist. I'm always wondering how Dave came up with this job title. From my training, I'm a remote sensing data scientist. I would claim myself. Okay. And I'm kind of an alien here in this conference. So you're looking at the microscope level and so on. I'm looking at a much broader scale, and I want to introduce you to my perspective today. And I'm going to talk about early detection and monitoring of wheat stem rust using very high resolution satellite imagery in Ethiopia. Dave had some of my work on one slide in his presentation in the morning, so I'm talking about half a slide from his presentation. And let's see. Okay, just to pick up where Dave left me, basically. So I'm in the context of the Disease Early Warning Advisory System, the DWAS project. And in this context, we want to develop remote sensing solutions for disease detection at a regional scale to improve disease scouting and disease surveillance. And basically, no one is really doing this with remote sensing data. So we kind of had to start from scratch using drone data, as you can see, in a field trial experiment. Then the next step would be going from the control environment on the plot field level, going to the field and the region level with satellite data. So for this, we are considering very high resolution satellite data. They have up to 50 centimeter spatial resolution. Here I mentioned two, like Planet and SkySat. And then the next step would be going to the regional subnational levels using a broader satellite, which is here Sentinel-2. It's for free. It's a 10 meter spatial resolution. So just to show you the pathway. When I started with this, I did a brief literature review looking at wheat diseases for detection and monitoring, using satellites for detection and monitoring. And I noticed that they focused only on 4 wheat diseases, actually. So yellow rust, or stripe rust, powdery mildew, FHB, and wheat streak mosaic. And when they did this, mostly looked at the grain filling stage. So this is not suitable for us in a context of disease early warning. It's always too late. It's suitable for damage assessment. And why they used, why the remote sensing community is looking mostly at stripe rust. It's clearly, you can see it by naked eye. So also the sensor can pick it easily up. But what about stem rust? You cannot see it so easily when the disease starts and is progressing and so on. So our challenge is basically in the early stage disease detection. We have no or mild symptoms at early stages. We have typically middle or to lowest levels of crop canopy where the infection starts. The next one would be when we move out from a controlled environment to a farmer fields to an uncontrolled. We have co-occurring multiple stresses, biotic nature. They have similar symptoms, so how can we separate these ones? And of course, we need high resolution data, especially in the temporal scale, where we can have a daily imagery. It increases our chance to get a cloud-free image during the agriculture season when we have a lot of rainfall. So, there were just basically 2 recent articles on stem rust using drones or satellites. And I mentioned just one. Okay, so the goal is to explore multispectral satellite data, very high resolution for rust detection. And so, the first question would be, do we have a reliable signal in the spectral data to identify the disease stress? Then the second would be, how early can we see this, at what severity levels, what's the genotype response, and what's the associated yield loss? Several groups are working with drone data. So the next question would be, can we use this drone data, the findings from drone database research to satellite? And then how can we replicate our findings in uncontrolled environments? How we handle biotic and abiotic stress? Unfortunately, we're not there yet. Luckily, we can work on that. So for this, we set up a field experiment in Debre Zeit in Ethiopia for stem rust early detection. Here's the experimental design. So we have basically 2 sites. They are the same design. We have 6 different varieties with a known to literature different reaction to stem rust. randomization, different treatments, and so on. The main difference between both sides is we have one is with a weekly irrigated scheme, and the other one is a bi-weekly. We did this during the off-season where we don't have usually rainfall, but my colleague Josef always says this is an unusual year, so we had rainfall. So the irrigation separation or this additional abiotic stress was not really working. We had only, I need to go up here. So we have only this time window where we had less rainfall. This is a time window of 18 days where this different irrigation scheme came into play. Okay, and important, we have 5 by 5 meter plots. It's not like a standard breeding plot size, so we had to scale it a little bit. So this is, I hope this works. Oh, damn.
It's on a slide. Try to just like this, right? Although I think someone else had a similar thing where they brought a video and it just didn't want to.
Okay, you can give it a click. No, super.
It's not clicking.
Anyway, I thought I used this slide, but basically, I mean, I wanted just to show how the satellite constellation is operating. So, I mean, they have like 23 nanosatellites. They're flying at the same time in different orbits around the globe and cover basically the same place on Earth on a sub-daily level. So, I mean, you can even have 2 up to 5 data sets per day from the same place. So this is the advantage of these kind of satellite constellations. Unfortunately, they are commercial, we have to pay for it. So there we need to think about how we scale this to a open source data. Okay, the SkySat satellite data has a spatial resolution of 50 centimeter, daily coverage or even sub-daily, and then we have like 4 spectral bands, the visible range, what we see, blue, green, red, and then the near infrared. And, okay, that's another one. Anyway, there's another slide missing about the data collection. So, I mean, we collected data, we did scoring on the ground on a weekly interval after inoculation. And we scored incidence, severity, and the host plant reaction. And then we created a disease index from this. And that's basically the disease scoring matrix we looked at. So, severity and the disease index, which is based on incidence multiplied by severity. Okay, now this slide is working. So from the spectral data, from satellite data, we used a spectral exploratory analysis using broadband physiological indices. So this is an approach which is commonly used in hyperspectral remote sensing. But basically we have 2 generic formulas. So this is a ratio spectral index. This is a normalized difference spectral index. This one is most widely known for the NDVI. So they use basically here the near infrared band, the red band, and then you calculate the NDVI. But we wanted to test more combinations of different spectral features or bands in this kind of context. And we also use some spectral indices, which is quite commonly known for disease. So for the SkySat satellite system, we calculated over 37 spectral features. And we tested this for early detection and monitoring purpose in a correlation analysis with the disease index per day, where we have the data sets. And also for phenotyping purposes, we calculate the area under the disease progression curve from the scoring, but also from the spectral features, the area under the curve. Okay. There are slides missing here. I don't know why. I mean, figures, actually. Shouldn't be like that.
Let's just try running it directly off this one, and then if you don't use the clicker, you just use these.
Okay. It's hard to show something without the figure, right? You can imagine this looks how it looks like, right? This is a satellite image. This is a disease progression.
It's so beautiful.
Just close your eyes.
No, I should have saved it as a thing. I should have done. And okay, so let's just quickly run to where you were. Yeah. Oh, so these are working now.
These are working.
Okay,
yeah.
so let's just not use the clicker and just use the laptop if that's okay.
Okay, then maybe just go back to the.
Where you were? I mean, sorry, it's yours now. So.
Ah, okay. Yeah. Okay, I see. Yeah. You want to see the satellites flying around again, or? here we go. Oh no, how do I...
Just give them go back to slides. And then
Yeah,
just
how do I start?
Let's just give it a click.
Let's see if they are. Yes. Yeah, no. I did this in the coffee break actually, so it's it should be here, right? Yeah, this gives you an impression of the system. Okay, yeah, now it's here again, my data collection slide. So yeah, we collect the data on a weekly interval. Maybe just give you 10 seconds, 5. Okay. Yeah, so let's look at the disease progression. So for the different sites, as I mentioned, site 1 was with the weekly irrigation, even with the time window where we had no rainfall. And you can see basically that the on site 1 we have like a faster disease progression and with more intensity, but you can basically notice, I mean, here day 30 days after inoculation, we have already here Hidasi, which picked it quite very well up. It's the most susceptible variety, but also the others picked up the stem rust. And here day 30, it's still quite low, the disease impact on this, of the same day basically. Okay, so basically looking at the disease progression, we need to split both, the data, the trials. And I created kind of a disease scenario based on the disease index. So where I would claim this one as like kind of a healthy condition here. We had some disease impact already on Hidasi here. So this is one variety which picked it up already. So there's some variation. Then I would call this slightly diseased and then moderately and severely. But here it was quicker, so we jumped directly to moderately diseased, severely, and then to the fully diseased one. So how does this look like in the data, in the satellite data? So, so this is on both sides, the healthy conditions, and then it's, okay, this one with the dashed line is the untreated block, and this is the treated block, and the same here. This is the untreated and the treated one. And I mean, what you can see clearly is from severely or here moderately, you get close to a fully damaged plots, and this is where the canopy looks different, right? So I mean, all these time steps is when the canopy is still green, something is happening in the leaves, some slight sclerosis and so on, but this is full necrosis here. So that's when the leaves are dry, dead. I mean, you know it better, right? So I mean, this is from a remote sensing perspective, it's like a clear change visually, but also like in the spectral signal, and that's what we picked up. But first, look at the yield loss. So I mean, so I mean, this is basically the experiment again with the different. I mean, this is the treated one. We have almost equally yield across the varieties for both sides, and then you can see like the yield loss. So for different varieties, and we have a good correlation with the disease progression and the yield as well at the plot level. So what I mentioned before, so this is just showing the most sensitive or significant related disease spectral features for the stem rust progression over time. And what you can see basically here, so this is for severity, for a disease index where we have only based incidence and severity, and this is incidence, severity, and the host response in there. And basically it doesn't matter which disease scoring metric we use, we can always find significant spectral indices. And for the early days, where we have basically the canopy is still green, so from day 17 to day 37, we notice that basically the blue band, for example in this combination here or here, plays an important role. And once the canopy switches to the senescent on full necrosis in the plots, the blue band is not important anymore, so we have here an offset to using the red band, for example. And usually what people do with remote sensing, when they do crop health assessment, they use the NDVI, for example, there's always based on the red and then the infrared band, but not on the blue. Blue is not considered. But they're looking always at the damaged crops. Or I mean, in the case of stripe rust, it's yellow, I mean, the surface, right? So this is kind of new. When you go to agri-tech companies, they're selling drones and sensors and so on. They take nowadays the blue band out. Blue is not an important band for agriculture applications, according to them. Now, we can see for STEMRAS at least, it's important, right? Yeah, so this is one of the main findings here. Yeah, and then if you look at the different disease scenarios, also across both sides, basically we can show like for, the healthy condition, this is basically biased for only one side because we had this little, here last we picked already a little bit disease up, so it's not really healthy in this sense. So we had the near infrared and the blue band as important bands here, and then for the mildly, also red, blue, blue, red, red, blue, blue, red in different formulas. For moderately, same, blue, green, green, blue, and so on. Severely, same, so they're repeating, and then for fully damaged is green, red, red, green, and so on. Yes. Yeah, and also looking at the disease progression, so it's maybe more for breeders interesting. It's basically the same. So we have also, again, the blue band, very important for associating basically spectral features to the disease progression over time. Yeah. Yes, and we're looking at the plot level, that's where the coefficient is at the 0.5, 0.6, 0.7 at maximum. If you go merging data to variety level, it's higher, but then we're introducing a mathematical thing. So, summary. So multispectral, very high resolution SkySat data seems very promising as a HTP, a high throughput phenotyping tool or early disease detection tool and monitoring for stem rust. So the blue region of the electromagnetic spectrum plays an important role for early detection in terms of low to moderate infection levels, but also for stem rust phenotyping. A next step would be like exploring now hyperspectral sensors for stem rust and FHB, early detection as well. And then, I have several these kind of experiments in Ethiopia. I think I have 4. And my idea is to train an AI algorithm on 4 different years with the different treatments. So one is like, we have different fungicide level applications. We have different varieties on early, mid, and late maturing, for example. Use this to train a model and then see if we can use this to detect biotic stress in pharma fields. But first on our own experiments and then in pharma fields. Yes, and thank you very much for all the institutions mentioned there, down there, for the support, collaboration. Yeah, and let's go forward with that one, right?
Super, thanks so much.